Ladies and gentlemen, boys and girls, good afternoon and welcome to Science of the Sabha. We are very happy to see all of you gathered here to spend the afternoon with us. My name is Ganesh. I'm a physicist and a faculty member at the Institute of Mathematical Sciences. On behalf of the Institute, I'm very happy to welcome all of you here today. We have an exciting evening for you. We have 4 talks bringing stories from the world of cutting-edge scientific research, especially research happening in India, that we'd like to share with you today. Before we start, I just have a couple of quick announcements. First, let me ask you to please turn off your mobile phones, but put them on silent preferably. If you do use data connections, you can use Facebook or Twitter to send us questions during the talks. Or if you're not on a mobile data connection, you could also send us your questions by SMS. Okay, so please do keep sending your questions through the talks. At the end of every talk, we'll have a few minutes for questions. We will have some selected questions answered from the stage here by our speakers. Even if you do have questions after the talks, then please do send them to us. At the end of the evening, after 4 talks, we will have some questions answered by all the speakers together. So please do send us your questions. We'll be very happy to answer them. The second announcement I wanted to make is we have an exhibition outside on Indian women in science. So today is the International Day of Women and Girls in Science, a day organized by the United Nations to honor the contributions of women and girls in science all across the world. So we have a small exhibition outside detailing the contributions of women researchers from all over India outside. So please do take a look as you step outside. On that note, let me introduce you the first speaker of this evening. So we have Professor Shubha Tole from TIFR Bombay, a distinguished scientist who will be giving the first talk today. So Professor Tole did her BSc from St. Xavier's College, Mumbai. Then she got her master's and her PhD degrees from Caltech. After a postdoctoral stint at the University of Chicago, she joined TIFR, the Tata Institute of Fundamental Research in Bombay, as a faculty member. She has been there since 1999. She has won many awards. She has won the Infosys Prize for Life Sciences, the Shanti Swarup Bhatnagar Award, the Wellcome and Trust Senior International Fellowship, and many others. She's a member of many scientific societies, the Indian Academy of Sciences, and many others. Perhaps what she'd like you all to know is she's also involved with Chai and Why, which is an event at TIFR Bombay on similar lines. They have a big gathering of people for scientific talks accessible to the public at large. So I invite her to the stage. You would have all received this little So you'd have received this little program sheet as you came in. So if you have it with you, please take it out. And if you've not folded it, please roll it out. Pretend you have a telescope with you. And Professor Shubha Tole will tell you what you can do with it now. Over to you. Thanks.
It's an honor and a privilege to be here. This is actually my first visit to Chennai. And to tread upon this hallowed ground, I'm treading lightly. Thank you so much for inviting me to this event. It's so nice to see so many people who are not actually practicing scientists come out of an interest in science. And I'm delighted to be able to share the science that I love. I study the brain. To me, there's been nothing more fascinating. To me, the study of the brain is a study of who we are and indeed how we are. My talk today is titled Outside In, How We Perceive the World. Our brain gives us a myriad of abilities. It enriches our life. I will make the case that it even makes our life. And perhaps I will encourage a dialogue where we can ponder what life is it actually making. Well, the world is full of beautiful things. I just have one picture, Music Academy. I thought I should first evoke music. I'm sure in this space you can surely hear the music coming from the piano. You can also see with great resolution the piano keys. Yeah? If we didn't have resolution, we wouldn't be able to see black-white keys. And certainly when the pianist plays, she's got to have resolution of touch. If our fingers were 5 times as fat, we would have had to make keys 5 times as fat, because otherwise we couldn't discriminate them. So this for me illustrates the senses which enrich our world. And what a beautiful world it is. I'm going to just show you some beautiful things and look at them with a neuroscience perspective. Flowers, what could be more beautiful? We all love flowers, yeah? And I'm sure we all agree that, you know, there's a purpley flower with white center and there's a yellow flower. Maybe some of you have these in your gardens. But you know what? This flower isn't necessarily purple and white and isn't necessarily yellow. If only our limited visual sense, if only we could see using wavelengths that bees use, if only we could see in ultraviolet, that's how you would see it. Okay, it makes sense, right? Look at this sort of centered kind of bullseye effect so the bees can find flowers. The flowers did not exist for you and me. We grow them. We're so proud of our gardens and, you know, we grow things. The flowers actually existed so bees can pollinate them. And the relevant signal is this, okay, which bees can see in ultraviolet. And we have been going along our merry lives thinking, oh yeah, this flower is yellow. It's not yellow. Actually, it isn't. It isn't anything. This is sort of a metaphor for life, at least to me. Nothing is, it's only how we perceive it. And this sort of describes life, doesn't it? Life is full of different people perceiving things differently. And there lies the whole Mahabharata. Everybody doing things as they perceive to be right. Sorry, neuroscience makes you a little philosophical. And I'm going to share this theme with you. You know, why study the brain? Well, it shows you how fascinating the world is that is sort of created in our brains. And it also shows you how little we actually know of the world. We live along our happy lives of yellow flowers. And hey, look, somebody else thinks they're red and white. Okay, this is sort of going to be the theme of my talk. These are flowers. Let's make it a little more scientific, you know, let's make it like boxes and so on. Yeah, that looks like it came out of a lab, right? These are our flowers. I'm sure people will agree that there is a sort of orange box with a brownish dot in the middle. Any disagreement? If you disagree, see me later. And then a sort of yellow box with an orangey center. Yeah, how many people are okay with that? That's okay, I'm okay with it too. I also see that. All right, let's connect the dots now. Ta-da, the centers are actually the same color, okay? If I removed the background, the centers are actually the same color, though even now, I at least see this side dot being a little browny and that side dot being a little orangey. I can't get away from that illusion. If I took out the centers, they'd be the same color. And this again allows me to sort of, you know, connect by metaphor. Things change with context. Everything we perceive changes with context, even color. Is it then surprising that everything in our lives changes with context? Why are our brains like this? How did these circuits arise? I actually study the development of these circuits because how these circuits grow allows them to function. See, our brain is a fantastic computer, but it's not one that you can assemble by buying parts. Okay, I have friends who can do this, I can't. But I have friends who can go buy motherboards and chips and literally wire them together and make computers. More power to them. But you know what? We all did this when we were embryos. We all made our own computers. We fabricated every single brain structure and we connected it up so that it would allow the brain and indeed us to function in this world. It would allow us to navigate this world. And what world are we talking about? Seeing is believing, we think. Let us see and let's see if we believe. Take your little rolls. Yeah. Hold it like so lightly with one hand. And the other hand is going to be in two positions. Position 1 and 2 at one end of the roll and the other end of the roll. Don't block the roll. Okay. It's just 1 and 2. Yeah. Now, keep both eyes open and just look into the distance and keep this in position 1 and raise it to your eyes. Keep both eyes open, okay? You don't need to squint. Keep both eyes open, and when you raise it to your eyes, one eye is going to have the hand in front, one eye will have the tube. Do you see me? Do you see me? Okay. And now move it to position 2. What do you see? What do you see? Position 2. Both eyes open. People of Chennai, what did you eat for lunch? You're saying there's a hole in the hand. Anybody don't see it? You can actually move it all down. You can make the hole travel everywhere. Can this be? You see it, right? I see it too. It's as real as anything. Look how the edges are stark. Look how the material inside shines. And the rest of it is your hand. What's going on? What's going on? Okay, I begin my graduate level neuroscience course with this demo. And I begin with this demo because this demo is based on fantastic visual circuitry. But first, I make my students debate. If we had time, I would ask you to debate, is the hole in the hand real or is it imaginary? And you should see there are strong voices on each side, okay? The point is, it's real and imaginary because friends, the whole world is imaginary. Nothing is out there. It's only what we construct in our heads. The hole in the hand is as real as anything. The reason we see the hole is that it comes from circuitry that allows us to see depth. Okay? This circuitry, you don't have a choice. Nerves from the two eyes connect to the brain in such a way that the images from the two eyes overlap. This is a mechanism to allow us to see depth. This mechanism is exploited when we go to 3D movies, right? We get these glasses. What those glasses are doing is literally one eye is seeing one thing, the other eye is seeing the other thing. You take out the glasses, you see blur because you're seeing two slightly shifted images. The brain knows how to overlap them. Isn't that fantastic? Isn't that fantastic? Yeah? Those 3D glasses are exploiting your brain circuitry, which grew. It wasn't hardwired. You weren't born this way. Okay, this circuitry of depth, the circuitry of overlap is wired up in your brain when we open our eyes and experience the world. When we open our eyes and experience a three-dimensional world where both the eyes are seeing two slightly shifted images, this circuitry wires up such that we can see depth. Isn't this fantastic? Our brains wire themselves up based on their experience of the world, in this case the visual world. They wire themselves up in order to help us navigate said world. Okay? Seeing has to be believing because after all, we gain information from the environment with our eyes. What else are we going to do? Okay? With our eyes, with our ears, with our senses. Let's examine this a little more. I don't know if you folks have decided whether there is a hole in your hand or there isn't a hole in your hand. But I think you might all agree that there certainly is a white square placed on 4 black circles. Everyone see that? Nice white square placed on 4 black circles. Okay. In fact, you can see, certainly this line you can see, high contrast. You can even see a line here, right? You can see a nice square. You can see perhaps a contrast difference between these two. Why else would you see a square, right? There has to be a contrast difference. But you know what? We're seeing things that don't exist. There is no line here, or here, or here, or here. This contrast difference is an illusion. Don't believe me? I'll block this out, and this out, and this out, and this out. There's nothing there. We're creating those lines. We're creating lines that don't exist because our brains are entrained to creating lines and making shapes. After all, they have to help us navigate the world, right? So our brains wire themselves up to create lines. This could be perhaps why children get so hooked on cartoons. It's all nice high contrast lines. Yeah. Here's another very cool one. This isn't even lines. I'm not going to make anything else appear. But just this set of lines like so on a plain white background. Do you all see nice white dots? Yeah? How many people see them? You can try and photograph. The camera is not going to get fooled. Only your brain will be. In fact, I don't even see dots. I see lovely diagonals running through. Yeah? Okay, all created for us by our brains. All created for us by our brains to allow us to navigate the world. The illusions don't stop here. Why am I showing you all this? These are not just funky illusions that exist in dots and dashes and lines. This is the circuitry that we use to perceive the world. Okay? It's not perfect, it's highly flawed, but it's what we have. And this is the circuitry with which we are so confident and so arrogant of our human, power. We are the highest evolved, right? We transform the earth. Yeah, we've trashed most of it, but we've transformed the earth. We've built big buildings. We've done so much. We have the biggest brains, and we're so proud of ourselves. There's nothing like neuroscience to make you really, really, really humble, okay? Because you see how utterly flawed your circuitry is. It doesn't stop with vision. But here's one more. Bisleri bottle, all of you had a good drink? Here is the Bisleri bottle in jail. Everybody agrees it's a Bisleri bottle. No, it was the same image. I just put some black bars in front of it. You've seen this image possibly through somebody's window somewhere. Now tell me, if a logical computer were to look at this image, would they conclude it's a Bisleri bottle behind bars? Isn't that slightly weird? Isn't the most simple explanation for this that there's a bit of the bottle there, then there's a black, then there's another bit of the bottle, then there's black, then there's a bit of the bottle. Computers have to be trained to be taught, boss, complete the image. Okay? Image completion is something our brains just figure out. And image completion is a survival instinct. Okay? Object completion. If we didn't have object completion, how would we realize that there is actually danger behind all these leaves? There's not bits of tiger behind trees. Okay? Now, the Bisleri bottle has become a tiger. Now you realize it's important to complete the image. Huh? Yeah, this circuitry works much better when the object is moving. Okay, when it's moving, the brain is able to trap bits of the image that all move together and complete it. Okay, you all have this experience. So I hope I'm communicating to you how our lives, our perceptions, how rich we think the world is completely ruled by our circuitry. Completely ruled. And then one must think, how much of the world are we not seeing? Okay, how much is there that we don't know about, and how much is there that maybe somebody else perceives differently? Okay, so it makes one realize that the sense of hubris that we have is actually just false, right? And it's not limited to vision. Everyone knows this, right? Ventriloquism. Yeah, it's just tricking your circuitry of sound. In fact, even though I don't have a puppet in my hand, I'm actually doing ventriloquism right now, right now. You guys are subject to monstrous auditory illusion. You're being manipulated. You know how? Where's my voice coming from? Speakers, where are the speakers? There. My voice is coming at you from wherever, and you're all lulled into the sense that I'm the one who's speaking. Why? Because my mouth is moving in perfect sync. Yeah, and that's how ventriloquism works. If I don't move my lips, and I have this thing in my hand, you're going to think that this thing is making the sound. Thank you. I can't tell you how hard I practiced that one. Okay. And here's a doozy. I tried to do this myself, but I just have to play this video. This is the McGurk effect. This video is thanks to Mark Milton, but the McGurk effect is thanks to McGurk. Now just watch and listen.
Ba ba, 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 ba ba.
Everyone heard that? that, right? Baby sounds, baba, dada, what that, yeah? Okay, that was the same soundtrack being played 3 times. Don't believe me? I'm now going to show you the 3 images of the person's face and the soundtrack playing. And I'd like to invite you to look at whichever face you want and hear what you hear. Yeah, sorry.
Ba 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 ba. Ba 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 ba.
So now we hear with our eyes. Okay? And now let's do touch. Anybody feeling confident and delighted and strong and powerful at being a human anymore? More power to you. Keep up the illusion. Okay, I don't have a funky experiment for touch. You have to imagine. Okay? I want you to imagine a tennis net. And I want you to close your eyes and poke at the net and answer this very challenging question. Close eyes, everybody. Poke, poke. Is this material solid or does it have holes? Answer? Did the experiment took the data? It has holes. Yeah, you're right. Prize for you. Okay. Now imagine this material. Open your eyes. Mosquito net. Take a good look if you've never seen one. Close your eyes and now poke, poke, poke. Does this material have holes? If yes, you've cheated or you have really thin fingers. Yeah. If I gave you a needle, you would surely see the material has holes, right? Okay, so we cross-reference one sense with the other, right? When I showed you the hole in the hand, you weren't willing to believe your eyes. You said, oh, well, look, I can't put my hand through it, so, you know, the hole can't be there. What probe did you use to cross-check? Your big, fat, if I may say, finger. Yeah? Hole in the hand, not there. Well, I asked you to do the same thing with the tennis net and mosquito net. Finger didn't work with the mosquito net, there you're using your eyes. It's just a matter of resolution, right? It's a matter of what resolution your probe has with which you probe the world. Okay? We navigate the world within the resolution we can see. Visual resolution, auditory resolution, there are sounds we can't hear. We all know that. Dogs can hear much higher sounds. Bees can see much higher wavelengths, higher frequency wavelengths. And touch, all we have is this, right? I'm going to now take you from this experience of, which I hope sort of perturbs you, it certainly perturbed me, to realize how little we actually know of the world around us, okay? If we were, 100 times bigger, we would not notice most of the, undulations of our territory. We would not notice many of the bumps, right? If we were 100 times smaller, an ant living on an orange would realize that the orange is full of mountains. Okay, so what is my conclusion as a neuroscientist? Nothing. It's really what we perceive, which then begins to plant the very perturbing idea that we're living in a matrix. I have to date not found an argument to persuade me otherwise. It allows one to not take oneself that seriously. Yeah, the next time one gets a heated argument, somebody cut you off in a lane, honking this, that, tumne ye kyun kaha and all that. Chill, boss. You're not really seeing much of what's out there. Why are you arguing about the little 2 bits you see? Yeah. Okay. So, how to build a brain? How does the embryo meet this challenge of creating circuitry that allows us to navigate in the world, or at least allows us enough of an illusion that we think we're in control of the world? That's what I study, and I'm going to give you a little glimmering of findings, data from my lab, to give you a sense of the excitement, to give you a sense of the thrill that your taxes fund. This excitement belongs to you. Please enjoy it. The challenges of building something complicated. Okay, these 2 movies are not from my lab, but I wanted to give you a sense, do you mind playing the first one, of what development is. Okay, this is a cartoon animation of human development. Start with 1 egg, quickly many. And there is the placenta. Let's bury ourselves in. This blue thing is going to become the embryo's nervous system, in fact, the body. Look at the entire nutrition machinery around it, the whole placenta. Even as the embryo is growing bigger and bigger and bigger, you have limbs popping out, eye developed, beginnings of the vertebral cord, spinal cord and vertebral column. Nice little arms. Now it's looking more like a baby. Placenta happily keeping it. It's a long process, huh? We all know it takes 9 months. And finally, a lovely new little human. Hey, quite a magical feat, okay? Quite a magical feat to produce this complexity. And now, let's see how much similarity there is in evolution, okay? Our embryos didn't invent this, they simply reutilized the mechanisms that they had learned in evolution. I'm going to show you what a fish does in a few days, okay? Everything, all of this that we take to do, much of it goes in developing placenta and developing nourishment and so on. A fish comes with its egg, packed nutrients, tuck, 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 everything is done. The most humbling creature to look at is the fish. It achieves in like a day, you know, sheer magic. And then I think, what did I do in one day? Can you play the fish video? So that's the yolk. That's 1 egg, 2, 4, 8, quickly, quickly, many. And now this ball of cells that's going to become the embryo will stretch over the yolk like a mask, see? Engulf it as it were. And slowly an embryo will develop in this crescent. It keeps its attachment to the yolk, just like the umbilical cord placenta. See there? That's the head, eye. See, beginnings of spinal cord, vertebral column, tail. But it's a fish, it can't grow curled up and sit in fetal position. So marvelously, the tail elongates, becomes nice and stretchy, becomes supported, and the head raises itself. And there you go, straight tail. It's almost doing yoga, no? Sarvangasana maybe. Yeah. And it's begun to twitch. Yeah, at about 5 days, the fish has responses, okay? In 5 days, it can start twitching. And it's continuing its development. And will soon become a happy larva that's ready to swim out in the world. At which point, it won't stick around under our microscope. It'll say, so long. Okay? So these are the magical creatures that develop their whole bodies. It's an incredibly complex act. What we study is how this magical system develops its brain. So that's a brain, and that is a simplified view of the visual circuit. Okay, this is not a lesson in neuroscience. This would terrify even neuroscience students. I just wanted to give you a sense of how complicated it is, okay? The signals from the eye navigate their way through to the back of the brain. From the back of the brain, they go through, this is a simplified map, And if I show you a bit of the cerebral cortex, this is what it looks like. This is a tiny millimeter piece of cerebral cortex where each color represents one layer of neurons. And if you can distinguish a neuron and its fiber, so this is one long wire coming out of one neuron, okay, and see how many there are. In fact, this is one such neuron in high mag, where each red dot is where some other neuron has come and connected to it. Okay, this connectivity problem is a nightmare. Our brains have 100 billion neurons, and each of them make between 1,000 and 10,000 connections. Okay, this is like asking, you know, the entire circuitry of, I don't know, a nano to be put into a nano box. I don't think this would be a problem an electrical engineer would like to take on, to make all of this circuitry for 100 billion neurons and then pack it into a skull, and then make it work so you can go navigate the world. And yet our embryos do it. All of us did this when we were embryos. Yeah? So here's how parts get assembled. Okay, this is a very short video clip from a friend and colleague, Kazunori Nakajima. I want you to see how brain cells move to where they have to go. Each red thing is a brain cell, newly born, and it's going to move and it's going to reach its location. This is a slice, a living brain slice of a mouse brain. Play it again, please. Very short, but you get a sense of how the cells are zipping around and somehow magically they form this control system that we know of as the brain. All this in the embryo so that our brains may form. How is each one programmed? How is each structure formed? Okay. So now, what did we actually do? This is a cartoon of a mouse embryo brain, and this part is the cortex where the mouse handles its sensation. And how does it handle its sensation? We're going to see how the circuitry of sensation forms. It handles its sensation because sensory nerves from all over the mouse's body reach a relay center in the middle, this, and from this relay center the nerves enter the cortex, and that's how the mouse can feel. So we have it too, okay? Now, if you wanted to feel and process sensation at high resolution, what would you do? Okay, if you wanted to take a photograph at very high resolution, you would simply go out and buy the highest megapixel camera you can, right? The more the pixels you devote to processing a signal, the better resolution you have. Now the brain has what it came with. So the brain does this very intelligently, if I may say. It says, well, there's a huge body whose sensations we have to process, but not all parts are as important. Let me devote larger bits of the brain to areas that we care about. And let me handle, areas where we don't really care about sensation. I mean, think about your feet, right? I mean, who slightly cares the small detail resolution? All you want to know is, are you walking on something pokey or not, right? But there are other parts where you really need to know. For example, your fingers. You really need to know. There are other areas that are very important. So the brain represents different areas of the body non-uniformly. It's not an equal opportunity sensory processor. So if I showed you the cerebral cortex, where I changed the size according to how much area each body part gets, here's what it would look like. Okay, the body surface in proportion. That's what we would look like. Yeah. So clearly, our hands are big sensory devices, and clearly mouth, okay, and kind of very little else. Yeah, you can kind of now see why babies explore the world like this. That's all they do. Yeah, they're taking information with the highest resolution sensors they have. Okay, now evolution has kept this mechanism intact. In fact, we learned it from our evolutionary ancestors. The mouse is the same way. Okay, for the mouse, the whiskers are the fingers. They're not just cute little things we draw on Mickey Mouse. Okay, when a mouse has to pass through a gap, it will actively whisk to see whether that gap is big enough for it to go through. It can actually move its whiskers and process signals from each whisker. Its whiskers are extremely important. That's the cortical map of a mouse. Okay, you can see it's pretty much all whiskers, all upper jaw, lower jaw, and four fingers. Not that different from us, huh? You suddenly feel close to the mouse now. No? Now, what is the circuitry, or what are the requirements of the circuitry of resolution? The requirements are that every signal has to be kept distinct. If I wanted to feel something complex, it's important that the signal from this finger and the signal from this and this all be separate in the brain. If the signals are merged, how am I going to be able to resolve anything? I might as well wear mittens, right? So big fat finger can become bigger, fatter finger then. For resolution, you need all the circuitry kept different. And I want to give you a taste of what data looks like. My wonderful student Ashwin found a gene that controls this circuitry of resolution in a mouse. Okay? When this gene is broken, the circuitry of resolution cannot form. And I'm just going to give you a flavor of what this data looks like. What is it that we get excited about? So in the brain, nerves from every whisker eventually connect to bits of brain like this called barrels. Now, why are they called barrels? Are there actually barrels in the brain? Well, the person who discovered them must really have liked beer because they were inspired by these images. And when you stain up a bit of the mouse's brain for a certain reagent, see, look, doesn't it kind of look like barrels? Yeah, now why are there so many barrels more compared to the whiskers? It's because there is fur all over the mouse's body. And all of these are representations of the upper jaw, the lower jaw, the forelimb, hind limb, tail, and so on. But most of it is given to the whiskers. And what my student Ashwin discovered, that's Ashwin, he discovered a gene which, if you break it only in the cortex, you don't break it in the nerves that are coming. Okay, if you break this gene in the nerves that are coming, it's like, you've broken the legs and now you're saying, oh, it can't walk, big deal, right? The nerves that are coming and carrying sensation are perfectly fine. It's only the cortex, the recipient territory, that does not have this gene, and the nerves aren't able to make barrels, which means this gene is really important for this circuitry of resolution. Okay, we are studying exactly how it does, but we have some theories. We think that this gene is necessary to make the cortex friendly to incoming nerves. And here's how we know this. I have just 2 more data slides, but they're exciting. So please share the fun with me. You've paid for it. This is actually a block of a mouse brain in which I've put in, this experiment was actually done by me. I'm delighted to be doing experiments in the lab as well as my students. I've put in, on both sides, a bit of dye. It's a lipophilic dye. It likes lipids, it likes fats. So it moves along the nerves whose insulation is coated with fat. And therefore we can track. We can track this pathway. And when we track this pathway, here's what it would look like, right? So here's where I put the dye, this big orange dot, and here's the pathway growing up into the cortex. And here's what it looks like in a normal mouse, okay? This is the pathway come in, and each little fiber makes a right-angled branch and goes up into the cortex and forms barrels. You can see these blobs? Yeah, we're not lying. They're actually like this. They're all clustered. Each barrel represents information from one whisker, okay? And one whisker is important to maintain resolution. So that's why this detail is important. Now look at what happens in the mutant. The pathway is normal, but the minute it hits the cortex, something bad happens. That's in high mag, okay? See, these are barrels, and here, where are those right angle branches? This thing doesn't even know what a right angle is, right? So it means that we are on top of the mechanism that allows incoming sensory nerves to enter cortex. And if it doesn't enter cortex, the mouse cannot process sensation. Okay, I'm going to leave this story here, but I wanted to convey something of the excitement of science. It's not something that lives in textbooks like a package. It's not something that's a product on a shelf that you buy. It's an ongoing living story. Okay, yesterday we discovered this gene. Today we found that, hey, when you break the gene in the cortex becomes hostile. The nerves can't enter it. Tomorrow we'll find out, why does the cortex become hostile? How do the nerves know to make those right angle turns? How does this affect the sensation and behavior of the animal? So that's where we're going. Now, I'm going to move to another sort of whole area of research, some of which we do in my lab. We process sensation from the world. What we process, we learn from, and what we learn from, we make memories. I'm privileged to be working on the memory machine of the brain, the hippocampus. It's not hippopotamus, okay? That's okay. Hippocampus. And this hippocampus records new memories, okay? It doesn't store them necessarily. The hippocampus is an unusual thing. It's an unusual structure because it is one of the few places in the brain where new neurons, new gray matter is being added even in adulthood. Most of the brain, or the neurons, are born when they are born. When the brain is in the embryo, it's all done. But in the hippocampus, one of the few places where new gray matter is added. So all of you here, tick, 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 neurons being added in your brain now. But not everyone's getting the same number of neurons. It depends on your environment experience. See this mouse, boring standard cage, and see its friends, lucky ones, in an enriched cage with toys to play with, stimulation. The first mouse gets some new neurons born in its hippocampus. These get many more. Not only do they get many more neurons, they also get neurons that sprout more, make more connections. Okay? Circuitry sprouts when the mouse is in an enriched, stimulated environment. And yes, this also applies to us. Slightly, we have to get off our butts and get ourselves moving and stimulated and do interesting things. Okay, we found a gene in the stem cells of the hippocampus that makes neurons where there weren't neurons before. How did we find this gene? We learned from the embryo. The embryo has stem cells, and we noticed that in these stem cells in the hippocampus, this gene is initially on, and when it's on, these stem cells produce neurons. This is a natural mechanism. And after a while, when the hippocampus has enough neurons, this gene gets turned off. And now, instead of making neurons, the same stem cell starts making a type of support cell. The support cell nourishes the neuron, insulates it, protects it, and so on. Isn't it cool? This lovely mechanism where one stem cell can make two completely different types of cells, all controlled by this one gene switch. Okay? My students, Lakshmi Subramanian and Anandita Sarkar, discovered that if this gene is permanently on, the stem cell always makes neuron after neuron after neuron, doesn't make a support cell. So we have a way now to add extra neurons in the brain that weren't there before. Now, we don't do this in all the stem cells because we need support cells, but we do it in just a few. And here's what a piece of the mouse hippocampus looks like before we created the extra neurons, without the extra neurons in the picture. And now I've overlaid the extra neurons in green. Look at them in high mag. Every green blob with that lovely arbor, it's almost like a tree. Every green entity is a neuron that shouldn't have been there in this mouse's hippocampus. But it's been produced because we kept this gene permanently on in the stem cells. Isn't this cool? Okay? We're able to put neurons in the mouse hippocampus that weren't there before. We're on the verge of finding out whether these neurons help the mouse make better memories. Okay, when I say better memories, I mean stronger, long-lasting, okay, not necessarily like happier memories. I don't know where happiness comes from. But isn't this again cool? This is, yesterday we discovered this genetic mechanism. Today we're putting extra neurons in the mouse hippocampus. Tomorrow we'll find out, do these neurons allow the mouse to make stronger, longer-lasting memories? This is again an ongoing process of science. This is why we scientists, our slightly mad-driven scientists, are every day is like this. Every day there is something new that we didn't know before. We couldn't think of a better profession to be in. So now, I'm going to give you a sense of what it actually means to grow a neuron. What it actually means when you learn anything. The neurons sprout and make more connections. All our learning is in these new connections. So to give a sense of what happens when these neurons are growing, sorry, a little bit of technical problem with the video, but this is a single hippocampal neuron in a culture dish, and it's alive. Boy, is it alive. Look at it. Look at its little tendrils coming out, sampling the environment, looking here, looking there, which way to grow. In fact, it's almost doing Bharatanatyam. Look. Yeah, even. It's dancing, it's seeking the environment, it's tasting. And then, all the sort of fibers on this side decide they're going to be the input fibers. They're going to take in input from the outside and bring it to the neuron. Whereas one of them says, I'm going to be the output wire. Let's watch it grow. One of them says, I'm going to go there. I'm going to sample the environment there. Now it's Kathak. I'm going to grow towards something. I see something there. I see a connection I wish to make. I'm going to connect. That's my target. That's where I'm going to grow. This is the circuitry of memory. This is the circuitry of thought. This is the circuitry of emotion. and it's growing in our brains. I'm going to close with a perhaps different idea, but one that's kind of special to me. And my study of circuits and how they grow sort of made me think, if our circuits grow based on experience, remember how I began with how the circuitry of depth perception is shaped and grown because of visual experience. Our circuitry is shaped by experience. When we learn things, we get new connections. We're able to mold our circuitry. And our circuitry controls not only what we experience, but what we do with it. Can we not then challenge our brains to growing circuits in directions we wish to grow in? Every difficult task, every tough assignment, every impossible skill that we try and master, and indeed every new idea, every new thought, every different person, individual lifestyle that we try to accept allows us to grow as people. And when we grow our circuits, if we grow as people, we also grow our society. What is society except a whole bunch of brains growing their circuits all together? Can we not then grow our circuits and our society to be more accepting, to be more inclusive, to be more tolerant? Circuits for society. Thank you very much.
Thank you very much for that exciting talk. Professor Tole is a student of dance, and she clearly is a natural on the stage of the Music Academy. We do hope to see you more often, perhaps in a different context here. We have lots of questions, so let me put a few questions to you now. So we have, so maybe there are two questions that are related. A question from the phone number ending in 834. Can the vision of a man be compared with that of a horse, for example? And there's another related question from Adityan. Do perception and illusion vary between species? So I thought this is an interesting context because most of your experiments are actually done on mice or on other organisms. How much can we learn about our own neural circuits from these?
Okay. Evolution has seen to it that the mechanisms that grow circuits in a mouse or indeed in a fly are the same mechanisms that grow circuits in us. So, when a neuron is trying to decide, is it going to be an input wire or is it going to be an output wire? I don't need to study it in a giraffe. I can study it in a Drosophila fly because the mechanisms that decide this switch are the same. So I can identify genes much better by choosing a model organism that's very tractable, such as a fruit fly or a worm. Evolution has seen to it that these mechanisms are the same, but the question was more of perception. Yeah, horse and human. Well, I'm going to answer, I'm not making fun of the question. I'm going to answer in a way that sounds funny, but that's to make a point. Can anybody ask a horse how it perceives? It's a challenge, right? I'm going to now move the question. Can anybody ask any other human what they perceive? Okay, this is straight into the realm of philosophy, so thank you, whoever asked the question. In fact, both are related. You know, the red that you see is not the red that I see and it's not the red that anybody else sees. There is no way we can communicate to each other what this feeling of redness is. Because what we perceive is based on the exact sensitivity of the photoreceptors in our eyes and the exact connectivity in our heads, which is slightly different for each of us. There is no way of actually communicating whether what you see is the same as what I see. And therein lies the joy of life and perhaps also all the conflicts of life. Yeah.
Thank you, thank you. Straight from the horse's mouth. So we go to the next question from Abhishek. The idea of using games to develop more neurons, neuron number or neuronal connections has been suggested, especially in the context of old people, to avoid degeneration or to improve memory. Is it really possible, or is it possible at the level of neurons?
If there's one thing that's absolutely clear about growing circuits, it's use it or lose it. Unused circuits will shrivel away, and challenged, stimulated circuits will grow and sprout. So, you know, we don't have a gene that we can put into our heads and suddenly get new neurons and so on. So we're going to have to do it the old-fashioned way. We're going to have to challenge ourselves with new things, new skills, hey, new ideas. And you know, one thing that really makes good new neurons, exercise. Gonna have to.
Okay, we'll take maybe one more question now. From Mr. Vijay Narasimhan. He asks, talking about resolution in the brain, people say that blind people develop better touch. Is that true? Is the brain redistributing its connections?
Fantastic question, thank you. The answer is yes, you already know the answer. Okay, the cortex is a processor. It will happily process input that comes in. In congenitally blind people, where their visual cortex at the back never ever got visual input, okay, the sensory cortex, the touch cortex, which is right next door, that sort of spreads over, and the connections that go to the sensory cortex spread, and they have more territory. More territory, more processing power. So absolutely, bang on.
Thank you very much. On that note, let's move to the next speaker. Let's thank Professor Shubha Tole again for a very nice, engaging talk.